before we dive into chemical reactions, chemical bonds, all of that fun stuff, I wanted to make sure we're all on the same page in regards to chemical formulas and how to read them and how to interpret them. Because we're going to use these chemical formulas and some models to represent different molecules and different types of bonds and things like that. So there's different types of chemical formulas. We'll in lab build some of these space filling models. We'll draw out some of these models that show us where the bonds are on that model, or we can just look at them as a molecular formula. Okay, but what we're looking at in these different types of chemical models is the number and different types of atoms that are in a molecule. So for example, if we look at this one right here, black is carbon, and all of these white pieces right here, those are all hydrogens. So you have one carbon, 1, 2, 3, 4 hydrogens in a single bond. There's only one line there. And then we can take this kind of structure and we can represent it with bonds. And now we have the C is the atomic symbol for carbon, and H is the atomic symbol for hydrogen. And so CH4, 1, 2, 3, 4, and they only have one line between them, so that means that they're sharing a single bond. Or even more simply, if we're just writing them out, CH4, carbon with 4 hydrogens. Okay, so that's how we represent different molecules. One of the key things being these lines here mean that there's a bond between those atoms in that to make that molecule. Okay, so let's read some more chemical formulas. So we talked about the subscripts. So the subscripts tell us the number of that atom that's in that specific molecule. So this molecule right here, H2, that means that there are 2 hydrogen atoms in this molecule right here. Same thing with O2, oxygen molecule. There are 2 atoms of oxygen in the oxygen molecule. The coefficients, the numbers that are not subscripts that typically go before the molecule, tell us how many of those molecules are part of that chemical reaction. So in this process where we're building water from hydrogen and oxygen, it takes 2 hydrogen molecules combined with 1 oxygen molecule. Okay, if there's no coefficient right here, you can just assume that it's 1 molecule. So 2 hydrogens, 1 oxygen gives us, when they combine, we get 2 water molecules. And each water molecule has 2 hydrogens and 1 oxygen. Again, if there's no subscript, you can just assume that there's a 1 there. So that's how we read the different parts of these chemical formulas and these chemical equations. So we've been looking at mostly molecular formulas now. So molecular formulas tell us the total number of each type of atom in that molecule, right? So molecule is multiple atoms bonded together. Here we have glucose, C6H12O6. That means that in one molecule of glucose, there will be 6 carbons, 12 hydrogens, and 6 oxygens. Okay, and the subscripts again tell us the number of atoms of each type in that molecule. We might see later in this class, or maybe as you move on in science and anatomy and physiology, what's called an empirical formula. So empirical formulas, basically what we've done is we've simplified the subscripts by dividing by the common denominator. So it's the simplest whole number ratio of the number of different atoms or ions that are in that compound. So we have, here was what we were looking at before, C6H12O6. What can we divide all of these by to get to the lowest common denominator? Right, we can divide all of these by 6. So 6 divided by 6 is 1, 12 divided by 6 is 2, 6 divided by 6 is 1, right? So it means that the empirical formula for glucose is CH2O, okay? This isn't representative of the total number of atoms in each of the molecules, but it gives us a kind of a shorthand way to represent the important components of the glucose molecule. Another thing that we're going to come across is this idea of isomers. It's a term that basically means the same. Iso means the same. And so these are molecules that have the same formula, but have a different arrangement of those atoms within the molecule. Okay, so again, black is carbon, white is hydrogen. These are different forms of the molecule pentane. So this is the molecule pentane. It's going to have 1, 2, 3, 4, 5 carbons and 12 hydrogens. If we count all of these hydrogens around here, that'd be 12. But these are all arranged slightly differently. They all have the same formula, C5H12, but they're arranged totally differently. So why does this matter? The difference in the structure of a molecule matters because that influences how living organisms can interact with that molecule. So for example, if this was like a sugar molecule, maybe our cells would be able to recognize this form of it right here, but the enzymes that break down sugar or interact with sugar in our cells might not be able to interact with it in this formation or this formation. Okay? Because the way enzymes work, enzymes are basically just molecules themselves, and they require a very specific shape of the molecule that they work on in order to function. So you change the shape, you're going to change its biological activity. That's why this is important. So same formula, different physical structure. Or we might have what's called spatial isomers. Okay, this one always, it takes me a long time to wrap my head around these spatial isomers. For me, because I'm not somebody that's really good at rotating things in my mind, that spatial awareness is just not really something I'm good at if I can only visualize it. I'm somebody that has to see it and physically move it in order to kind of grasp this idea. And so that's one of the things that we're going to be doing in lab. So for example, here we have carbon group with a carboxyl group with an R group, which is part of an amino acid. This can be many different shapes, an amino group and a hydrogen. Okay, so these two molecules have the exact same parts to them, right? But they're kind of like left and right hands, right? They're mirror images of each other. We could face them to each other and they would be mirror images of each other, which is what we're looking at right now. These two molecules are mirror images of each other. But we cannot take these molecules and rotate them together in any way that they would fit directly on top of each other. And they could basically sink one into the other and be an exact copy. So the relative orientation of the atoms are different. And again, this influences how organisms and how their enzymes and how these molecules will function within cells based on the orientation of those different parts of that molecule.